Ready to talk about synapses. So this is one neuron talking to another, opposed to signaling just down a single axon, a single nerve. So what I want you to start with is actually draw yourself another neuron and label the key structures of your neuron, and also include where chemically gated channels and voltage gated channels are present. I've done this before. You should be able to do it now. Okay, this is what you should have come up with. We've got the parts of the cell, axon terminals are here, and then the key gated ion channels are the chemically gated along the cell body and dendrites, the voltage gated sodium potassium channels along the axon, and the voltage gated calcium channels at the axon terminals. This is what we're going to be using more now. You should know the functions of all the other gated ion channels already. So this neuron is going to be talking to either another cell, and we're going to look at right now is that being another neuron. The other terminology that I've mentioned before is this would be the presynaptic neuron, and this one, the second neuron, is the postsynaptic neuron. So let's look at just those two neurons in a little more basic here. Our presynaptic neuron is talking to our postsynaptic neuron, and there's two broad ways that it can do this. It can do this electrically, or it can do this chemically. We're going to be focusing on chemically, but I want to briefly tell you about the electrically as well. So here are the two different types, and the differences in anatomy here is not that important besides this gap. So here is our electrical synapse, and here is our chemical synapse over here. And the only difference in anatomy that I want you to notice is this gap right here is a synaptic cleft. It's a gap between the two cells. So I do want to tell you just a little bit about the electrical synapse. This is basically your gap junction. This is what you've seen before in terms of junctions between epithelial cells. Is basically a protein that connects these. I'm just going to color over this because it's then a protein that allows these two cells to be connected electrically. So when you have a depolarization in cell 1, that is going to allow ions, so let's say this is sodium, right, flowing. Those ions are going to flow right into the adjacent cell. So it's electrically coupled. Ion movement in cell 1 creates ion movement in cell 2. And we'll see this type of synapse again, type of connection with cardiac muscle. This is how your heart contracts all at once. We're going to be focusing on chemical synapses today. This is where the presynaptic neuron and the postsynaptic neuron are separated by a cleft. This cleft here is a gap, and the synapse refers to this entire kind of junction here. A chemical synapse means that it's going to release a chemical messenger. This is going to be called a neurotransmitter, NT. I'll abbreviate that for now. That's going to then bind to, guess what? A ligand-gated ion channel on the postsynaptic membrane. Okay, so let's look at the chemical synapse in more detail. Here is a chemical synapse. We've got synaptic vesicles that are at the end of the synaptic terminal. So here is our synaptic terminal here. We're zoomed into one. Sodium potassium channels all along the axon, but then at the end of the axon terminal, we've got these calcium channels instead. These calcium channels are going to allow calcium to flow into the cell when there's a stimulus. What might stimulate these calcium channels to open? Well, they're voltage gated. So a change in voltage, depolarization. So this action potential traveling down here is going to cause these voltage gated calcium channels to open, and calcium is going to flow into the cell. What does that mean about calcium concentration inside and outside of the cell? There must be more outside. And I told you, I think last week, that there was high calcium outside the cell. This means that calcium will flow in when those channels are opened. The flowing of calcium inside the cell causes synaptic vesicles to fuse with the membrane and causes the release of neurotransmitters that are just hanging out there inside the cell. So these vesicles are hanging out here, but they're membrane bound, and they're going to be waiting for the signal and then be released via exocytosis. This is going to cause neurotransmitter to bind to a ligand or chemically gated ion channel on the other cell and then cause something to happen over here. We'll get back to that. So here's some of the terminology here. Synapse is this entire connection. Synaptic cleft is the gap. So cleft refers to the gap. And then the synaptic vesicles are full of neurotransmitter. Okay, we are going to go right here and walk through this process. So here's our chemical synapse, presynaptic membrane, postsynaptic membrane, cleft. We've got high calcium outside the cell. We've got our synaptic vesicles. And we've got our voltage-gated calcium channels. So when an action potential comes down and reaches the axon terminal, it's going to cause voltage-gated calcium channels to open and calcium to flow into the cell. When calcium flows into the cell, it binds to these vesicles and causes them to fuse with the postsynaptic membrane. Like that, boom. So this neurotransmitter here, I can label that neurotransmitter, is released into the synaptic cleft. The neurotransmitter is going to bind to the chemically gated channels and cause them probably to open, and then something to happen here. We'll get back to that. It could either be excitatory or inhibitory. Is this an action potential necessarily? No, but it might be a graded potential, either in a depolarizing or a hyperpolarizing direction. So for example, one example, we'll come back to more of these. Glutamate is a neurotransmitter that binds to chemically gated sodium channels. When sodium channels open, sodium will flow in and depolarize the cell. That may or may not be sufficient to reach threshold.